I would like to introduce Dr. Simon Dinek, University of Zurich, and he's going to talk about SARS-CoV-2 neuronal infection.
Good morning. So I will show you our data, which is looking at the infection of the brain of mouse with SARS-CoV-2. So why we do look at the brain is because if we look at acute COVID-19, if we look at the long COVID form, we have a frequent report of neurological signs in patients. And although it's quite frequently reported, we don't really know which are the mechanisms. So at some point, quite early, I would say, we thought during the pandemic that there could be this direct brain invasion, which would lead to the neurological sign. Since then, several reviews have shown that this is quite a rare mechanism. Some show still that there are some particle or piece of virus within the brain, but still to be something less, not on the top of the list to explain it. And then if we look in long COVID, there seem to be also additional factors which could explain it. Some of the studies show a psychological component. So what we want to do here with the mice is we want to look at what's happening in the neuron, if there are any morphological alteration, and then want to try to compare that with all the molecular data we can gather to see if they can add together to give us a better explanation. So the hypothesis for this work is that we have SARS-CoV-2, it infects the neuron, but it does not lead to a cytopathic effect, although we would have an innate immune response. So for that study, we used the humanized mice, so the K18H2, we infect them with SARS-CoV-2 Delta 10 to the 2, 6 to 7 days post-infection, we collect the brain. And so we do transverse section here, level I, hypothalamus, thalamus, and that we use for omic data. So we do lipidomic, metabolomic, we do the transcriptomic data, and then we also look at ultrastructural section. For the rest of the brain, we take the frontal cortex, we confirm infection with PCR, and then for all the rest, we just look at the microscope and we try to see if we see any changes. And one brain is used fully for light sheet microscopy. So that's what you can see here. So on the left, you have transverse section from top to bottom. On the right, you have 3D reconstruction. And so what you're looking at here is in yellow, orange, you have the SARS-CoV-2 nucleocapsid protein, which is marked. And so you see that it's mostly located at the level of the cortex, mostly on the surface, and at the ventral aspect. And so this is what we would call a widespread infection. So we have a lot of positive neuron. But although that's the most common one we have in the study, you will see, we have also other patterns. So now we have a brain of a mouse, it's cut right in the middle. So you have this longitudinal section. And so one pattern we can have is the scatter one. We have very few positive cell process. Then we can have a bit more. So we have this cluster of positive neuron. And then the last one, which is all the case within this study, and that's what we just saw in the light chain microscopy, is what we call the widespread one. You have a lot of positive neurons. So here we look at the nucleocapsid protein. Now, so again, on the right, immunohistochemistry. On the left, you have our classical stain, so hematoxylin and eosin. And you can see that the neuron, so pointed on the right, the brown one with the arrow, are completely full of the nucleocapsid protein. But if we look on the left, it's consecutive section, you see that we have the neuron as well. But for us, they look completely fine. So if we just look at this, we don't see anything wrong going on. So we can also confirm here nucleocapsid, again, we try with the spike protein this time, and also the neuron are completely full with it. So we are a bit puzzled because although the neuron seems fine, there are a lot of viral material in there. So we think, okay, it would be strange that there is nothing at all happening. So what we do is we go for the transmission electron microscopy. So on the left here, neurons. So in blue, you have the cytoplasm, which is delimited. In the center, you have the nucleus, in orange, delimitation, and then you have several organelles. In green, you have the mitochondria. In red, you have the endoplasmic reticulum, and in yellow, you have this accumulation, which could be lipofuscin. And so overall, you see that the cytoplasm is busy, but have this kind of regular organization. Now, if we look in an infected one, you see that there is something more here, which is added, and those are the viral particles. So you see that you have a membrane here, and it is surrounding several of these round particles, which are SARS-CoV-2 viral particle. So this is an infected neuron. And if we go, so here you have back the cycle of SARS-CoV-2. If we put it together here, it's a different section. The difference also is this time we do immunogold. So we take the antibody against SARS-CoV-2, we put a little gold particle on it, and which is going to appear as a black dot on the electron microscopy. And so you see that the black dot here are fitting with what we called before the viral particle, which is which make our assumption stronger that this is indeed the virus. And so our assumption so far is if we try to link what we see with what data is showing how the cycle would go, we think that we have at this step. So we have the virus which is forming the neuron and which are ready to be released. And then we have those very large accumulation here of black material, which is also, you can see we have this little dot. So there is nucleocapsid protein. So when we try to think what it could be, our hypothesis now is it would be this step, that we have this accumulation of viral material within cytoplasm, and that it would appear like this morphologically. Another thing that we did to confirm it is we did in vitro work, and so we use this HAK cell that we put ACE2 receptor on top of it. We infect them with the virus and we do again gold staining for nucleocapsid protein. And you can see that first of all, morphologically, we have those similar structure and they're also marked with the antibody. So which is also make us stronger our statement that is very likely to be the virus. And then what we try to do now is we try to see if there is any difference in the transcriptomic data. So on the left, on the PCA, you can see that we have very nice clustering of the brain. So all the infected one are on the right in orange, and all the mock one are on the left in green. And if we look at the marker that the software used to cluster them, it mostly uses here chemokine factors. So you see CXCL9, CXCL10, etc. And if we look now on the volcano plot for the differential expression, what we see in the affected brain is mostly an upregulation of gene. And again, we see this mostly having to do with inflammation. Now, if we look at the top 10 pathways, we did GO, BP, and we did KEGG. Most of them have to do, if we look at the top 20 unbiased, have to do with inflammation or innate immune response against the virus. Then we try to dig a bit in more detail, because what we saw on histology is that we have a neuroinflammation, which is characterized by the recruitment of leukocytes. So we have the leukocyte in the brain, and so we can see on the left that we have inflammatory mediator, which show that there is indeed activation of all the inflammatory pathway, which would make sense with what we see. And also we have the presence of pathway which are also positively enriched associated with leukocyte, so which here also fit with what you see morphologically, so that there is a neuroinflammation and that we have the recruitment and activation of leukocyte within the brain. Then we try to look in the innate immunity because we have the infected neuron, and so we somehow think that the brain should sense their presence. And again, if we look at this different pathway, all of them are positively enriched. So from the receptor to the interferon response. And then if you look at the last one, the reaction, we have again here enrichment of several pathways which have to do with cell response or body response against a viral infection. Interestingly, there is not only inflammation or innate response, we also have change in pathway which have to do with neurons. So for example, a bit more morphologically related pathway, let's say, we have this axoneuron projection, neuron morphology pathway. Most of them are negatively enriched. Only one of it is positively enriched. Then we have also change at the synapse. Most of them, again, are negatively enriched. And then we have also change at the level of the behavior. And so we see something like walking behavior, social behavior, exploratory, fear response, they're all negatively enriched. And so we have collaborators in Liverpool who look a bit at some behavioral tests in the mice, and they could also see somehow some difference between the infected one, which are in red, and the non-infected one, which are in blue. So how we can correlate the two, for now it's still a bit unclear, and we need to look a bit more into the detail, but somehow there seems to be something happening in the mice. Now, because we have bulk RNA sequencing, we don't really know what's happening at the cell level. And so we try with the cell deconvolution to approximate which cell type we could have within our sample of thalamus and to see if there could be a difference between the two types. And so as you can see here, so we look at the mock infected one here, we look at the delta infected on the left, and most of the cell type here are glial cell. We don't see a lot of difference, except here you see the red one would be the microglia. And you see that the proportion here in the infected is markedly enlarged by comparison by the uninfected one. And then you have here in this dark blue, this is inhibitory neuron, and it's pale beige, you have the excitatory neuron. And you also see that there's a difference in proportion. And so we're still investigating what it means, if this is something real or just because we have a shift in proportion of some microglia that it will impact also the other proportion. But regarding the microglia, that's something which I'm not very surprised because we have already reported in our group that there is microglioma, microglia activation in infected brain. So that one we're quite confident that it's something real. Now we need to find out if the neuronal change are also something real. Then we also try to look at lipidomic metabolomic data. And so our colleague, which are trying to prepare this analysis, first look at the protein content to see how they could normalize. And so this is something we have no explanation for at the moment. So we have here 18 groups. The one we're interested in today are group number 2 and group number 6, so the delta and the mock. And you see that there is a large variation of protein content between the sample. We've tried to explain it by the PCR level to see maybe if there is more virus, there would be a higher protein content. But as you can see here, when we do this correlation analysis for group 2, we don't really have a nice correlation. So we are not really able to explain it. And we don't really have a clear explanation why we have such a variation. And so far, we just have some preliminary data for lipidomic and metabolomic. So you can see that it's a bit the same shape as the volcano plot for the transcriptomic data. So we have a lot, which is a shift to the right. So seem to be a lot of upregulation or increase amount of those metabolic lipidomic, which is significant. Preliminary results so far, we can see that we have an increased pentobarbital, corticosterone, beta-hydroxybutyrate in the infected brain compared to the mock infected. So of course, we need to look into more detail, but this is just what we have so far as preliminary data. So if we try to write it up, our hypothesis so far is that we have an infection in the neuron. This is going to trigger the innate immune response. And we don't know how yet, but this would also lead to some change at the level of the synapse, also some change at the level of the behavior. And beside that, of course, we have this innate immune response or this infection, which is going to lead to a neuroinflammation by what we suspect to be an astrocyte and macroglia activation, which are going to lead to the creation of a local pro-inflammatory environment with the recruitment of leukocyte. So that's something we already done before if you're interested. And so with that, I would like to thank you, all our colleagues, including the one in Liverpool, which helped us with the experiment. Thank the funding body. I also thank you all for your attention.
Thank you so much, Simon. Any questions?
There's one at the front here.
Hi, so that was a really great talk. So what I noticed in your data is the type of things you've observed with the microglia upregulation, the neuroinflammation, everything before you presented your lipidomics data. I know it's preliminary data, but have you looked for any lipid metabolism dysregulation that kind of mirrors other neuroinflammatory and other neurodegenerative diseases? Because all the other data you've presented kind of mirrors things like Alzheimer's. So could you comment on how SARS-CoV-2 infection might be affecting those, the development of those other conditions?
Okay, thank you. No, that's very interesting. We didn't know it's just the primary. So of course we need to go a bit more in the lipidomic metabolomic. When we look at the transcriptomic data, we had some already indication that there might be a change in some lipid pathway and metabolite pathway. So that will take in account. Indeed, it would be interesting to compare with other diseases to see if we can match some changes. I know that when we look at the transcriptomic, we often have this Alzheimer's disease coming up, like some paper showing it Newman, and then you try to map some gene and you see some difference. So that's indeed something we should look into.
Thank you.
I think we should round up there. I hope you all noticed just how exquisite some of the microscopy was in this talk, which is quite unusual to see in a virology conference, and really complemented an excellent talk. Thank you very much.
Thank you.